Hi, everyone. Thank you for joining us today for a webinar on maximizing efficiency with a dual DNA and RNA purification solution from the same sample. My name is Claire McLean, and I'm a marketing associate here at Omega Bio-tek. Joining me today is Nelson Cottram, one of our field application scientists, as well as Caleb Proctor, the research and applications manager with Omni International, a Revity brand. On the agenda for today, I will start off with a brief introduction to Omega Bio-tek before handing off to Caleb for an introduction to homogenization, as well as a review of some data they've collected. From there, Nelson will explore the automation capabilities of this workflow before showcasing some performance data. Finally, we will end today's session with a Q&A, so make sure you submit your questions below. With that, I would like to introduce you to Omega Bio-tek. We were founded in 1998 and are based out of Atlanta, Georgia. Our expertise is in DNA and RNA purification solutions, and we boast a portfolio of over 400 products encompassing different extraction technologies, including magnetic beads, silica spin columns, filter plates, and salt-out methods. Omega Bio-tek is ISO 9001 and 13485 certified, and our presence is felt around the globe through more than 50 distribution partners. At Omega Bio-tek, our purpose is to deliver nucleic acid purification solutions that simplify your project and meet your unique needs. Your success is our top priority, and we empower our customers by making automation and quality products more accessible and affordable. With that, I will hand you off to Caleb from Omni.
Thanks, Claire. Hi, I'm Caleb Proctor. I'm the Research and Applications Manager here at Omni International. I work for Revity, who is a large global life science solutions and service provider. I specifically work for the Omni line of products, and I support the homogenizers that we provide for various laboratories across the life science industries. Our mission is to empower scientists and focus on discovery by innovation and simplifying sample prep workflows. So when it comes to homogenization, not every lab needs homogenization. Some samples that you get in are liquid or pretty easy to lyse, like cell culture. But in a lot of situations, labs will work with very esoteric samples or in models like mice or ferrets or something like that, where you'll be working inside of a tissue and you need to extract an analyte out of that tissue. So generally, a lab implements homogenization for one of these four core principles. To start with, normally a lab is looking to speed up a process where they're working with a very time-consuming enzymatic digestion or a chemical lysis, or even some very lower throughput manual methods like mortar and pestle or a Dounce. A lab may also want to increase the yields of these target analytes inside of tissues or solid matrices by getting a complete lysis of these cellular matrices and pushing all those analytes into solution. Finally, it may be a QC step that they want to implement where they have programmable methods to reduce sample-to-sample variance and reduce user error inside of a workflow to have a very cohesive and tight, reproducible workflow. And finally, the last piece of homogenization is that it will provide innovative ways to extract analytes from these solid samples that may not otherwise have been used in past workflows. Maybe a researcher is using a enzymatic step in a protein workflow where they do not want to introduce various other proteins inside of that, and you can remove the enzymatic part and just introduce physical lysis to lyse all these cells. Or maybe they don't want to have extra chemicals, extra STSs inside of a reaction, and this single step will provide a way to get all of these analytes into solution that they otherwise did not have. So when it comes to homogenizing a sample, I like to say that we can homogenize everything from bones to bacteria. And when I say that, I mean everything from a mouse bone, a cow bone, all the way to the very tough microorganisms that you're working with. We can get it all, and everything in between, from plants to stool to the dirt under your feet. It can all be put inside of a tube, and we can help get these analytes into suspension. We work with a variety of sample sizes and a wide variety of buffers, everything from acetonitrile, methanol to the common lysis buffers that you'll find inside of a kit. It's very compatible with that. So we take this wide variety of sample types, and we're also able to work inside of a wide variety of various common laboratory consumables things that you're used to seeing on your bench tops, like 2 ml tubes, 50 ml tubes, 15 ml tubes, well plates, to even some more specialized tubes like 30 ml tubes, 0.5 ml tubes. The list kind of gets comprehensive, but many different labs are working with different types of plastics, and we try to fit that inside of a workflow that'll work for them. The lysis that we provide is buffer agnostic, physical lysis. So you can homogenize a sample in something as simple as water, or you could even homogenize them in the absence of a aqueous media in total. Just whatever generally will fit inside your workflow. We try to accommodate that with our devices. Uh, all these samples, no researcher is just looking at homogenizing for the sake of homogenizing. There's always an analyte that they're after. I generally narrow that down into DNA, RNA, proteins, lipids, small molecules, elemental analytes. We take this huge universe of samples, and we're really looking for those single, those single pieces out of that. Those samples feed a variety of workflows, whether that be nucleic acid extractions or total protein analysis. And the list goes on and on as far as what analyte analysis that you're trying to do. If you're working inside of a solid sample, our homogenizers can fit on top of all of that and provide the consistency and the speed and the yields that you're looking for to maximize the throughput inside of your lab. Today, I want to talk specifically about bead eruptors. We offer various other homogenizers, including rotor-stator technology and sonicator technology. But in this specific application, I want to highlight the bead mills that we provide. To start with the Bead Eruptor Elite, it's kind of our most varied sample. It accommodates all those tubes I just spoke about, from 2 ml tubes to 50 ml tubes, and everything in between. We can put everything from bones to bacteria inside this. We have applications featuring mice bones and applications that will cover those tougher to lyse microorganisms as well. All of our sample disruption takes place in less than a minute for most samples, and then all the way up to around 5 or 6 minutes for even the most toughest samples. We can really lyse all this up and have it done quickly in sample numbers of 1 all the way to 48 with the 2 ml tubes. The Bead Eruptor 96, which is the instrument that we feature in this webinar for the DNA extraction and RNA extraction that we do further, is a more specialized device. Typically, we want to work with either milling jars or well plates inside of this device. The milling jars typically will dip in liquid nitrogen, and they can generally homogenize anything you put inside of it. We've homogenized rocks, PC boards, big cow bones, things like that inside of a milling jar when you really want to powder something that's incredibly tough. But where it shines for this specific application is in a high throughput environment where you want to use 96 deep well plates and a solid matrix to feed into various other extraction pipelines. It's ideal for tissue homogenization and incredibly tough matrices, like I said. But we have a wide variety of applications to support everything, including DNA and RNA extractions. To get into the topic at hand, I wanted to paint the picture for what we built this application around. We initially started with a customer who wanted to work with perhaps a wide variety of samples in a high throughput setting, which would obligate you to use something like the Bead Eruptor 96 for well plate environments. The kit itself that we chose, the Omega Magbind DNA and RNA kit, begins with a single sample where you can get isolated DNA and isolated RNA from a common source. And this is pretty unique when it comes to DNA and RNA extractions, because generally DNA and RNA extractions are a single pipeline where we use nucleases to remove DNA or RNA from each of the extractions. We find that this works really well whenever you're working with small and precious samples that otherwise may not be able to be subdivided, and maybe you would have better outcomes if you could take these small samples and create extra replicates to make duplicates or triplicates of the same sample to sort of increase the integrity of the data downstream. So what we started with was fresh mice organs for this. We took liver, heart, kidney, brain, and skin samples, and we divided those into many different 5 and 10 milligram plus or minus 1 milligram samples. We loaded those into a pre-filled 96 deep well plate that contained 1.4 millimeter ceramic beads. This is a general tissue homogenizing mix that we have for the Bead-Raptor 96. And inside of each of these wells, we had 300 microliters of the lysis buffer from the Omega Magbine DNA and RNA kit. We put each of these well plates on the Bead-Raptor 96 and homogenized them at 30 hertz for 1 minute for all of the liver, kidney, heart, and brain samples. And then we additionally homogenized the skin samples for 1.5 minutes. They're a little tougher, so they take an extra 30 seconds to fully homogenize. We took all these homogenates, we transferred them to a centrifuge, and we centrifuged them for 10 minutes at 10,000 times G. And then we transferred all of those lysates into tubes and then proceeded to extract both DNA and RNA from each of these individual samples using a very manual extraction. The beauty of the kit that we chose is that magnetic beads are flexible for both manual and automated pipelines. So you may be working with a lower throughput extraction set where you want to work with a few samples, but those same few samples will immediately be transferable into a high throughput scenario on something like a liquid handler. We took all the DNA and RNA extracted from this manual extraction on our end, and we submitted them to analysis with a nanodrop, a tape station, and a Agilent 2100 bioanalyzer, so that we would have not only have quantification scores for 260 over 230s, but also have concentration and RIN and DIN values for both DNA and RNA. The QC that we used for all of these nucleic acids, we used from a third party and their QC criteria for their core lab. This lab does thousands and thousands of samples, and this is on their input worksheet where they require these minimum thresholds to meet criteria for 95% effective analysis for both DNA and RNA, transcriptomic and genomic workflows. We find that using this third party is a really good minimum QC criteria for all these various DNA and RNA extractions because of the varied amounts of samples that they get in. So let's get into the DNA extraction results here. So with really good results for both 5 and 10 milligram samples across the board. The liver, kidney, brain, heart samples, both 5 and 10 milligram samples, all met the QC minimum thresholds that we established in the last slide. The highest concentration we received from the 10 milligram samples, and the lowest being from skin samples in total. I will also point to the fact that the skin samples did not meet the QC criteria for the 260 over 230s. But we generally don't see that as an issue because with other kits, we can add an extra wash step to sort of increase this amount. But we wanted to show everything with a single pass for the DNA and the RNA just because of how well all the other matrices work. For skin specifically, with it being an external organ, you're going to get a lot of other contaminants on the skin that might come into your DNA extraction. Typically with 260 over 230s, you'll see these lower scores and absorbance show up from added salts, added ethanol, or lipids that are passing through into your elution. As you can see from the other extractions that were done on the same kit in the same way, their 260 over 230s were fine. So that would generally lean to the fact that there's no ethanol or salts making it through from the kit itself. I would say that on the top end, is likely that these are just a lipid contamination that we can wash out. But I just wanted to point to that as the only outlier inside this entire experiment. The rest of it, everything looks perfect and came through our QC criteria as acceptable. For the capillary electrophoresis analysis, what you see here is a fragment chart from the tape station. We've displayed all of the liver, kidney, brain, heart, and skin samples in a row there for you. As you can see, the fragments generated are very consistent across all the sample types that you see here. Homogenization, physical lysis, is inherently going to shear DNA. I mean, DNA is very fragile. You can shear it by pipetting. So it's well known that this is going to affect the fragment size of DNA. There's just no way around it. But the good news is that this is a very repeatable process because these programmable methods allow for that consistency to take place. Across all the samples excluding skin with DNA, we range from 13,000 base pairs all the way to like 16,500 base pairs. This was including the fact that these samples were 5 milligram and 10 milligram samples. But you can see inside the common samples, the liver, specifically the kidney, etc., that the range of the base pairs is only 1,500 plus or minus on across the board. So what that really speaks to is just how consistent each individual sample can be. Now going back to the skin here, because we had to homogenize it for 50% longer, you can see that the shear is getting worse if you're worried about shear. That you're getting 10,000 or 9,000 base pairs on these samples that you see here. And however, all of these still fit inside of our QC criteria. The only reason I bring this up is that there's some labs that may be interested in maintaining a very high fragment size to fit inside of maybe a long read sequencing workflow. But what this displays is that we can really get this very consistent fragment size if you are worried about that and want to program around that and build that into your QC criteria, that we can absolutely fit that. And maybe you don't even need to homogenize as long if you're working with a single sample. You can really dial in your extraction criteria to meet some of these size parameters. But everything here fits inside the QC and everything looks very nice. When it comes to the RNA extraction results, everything looked great. We had between the 5 and 10 milligram samples for liver, kidney, heart, and brain. We had very scalable amounts of concentrations for the RNA yield. The liver itself had 25 and it scaled into 43.5. That's roughly 2x. So that is a good sign that this is scaling well across the replicates. The same can be said for kidney, brain, and heart. A skin here was a little more difficult. It didn't want to produce the same good results that we saw, but it did meet the QC criteria that is listed in the table. As you can see, the concentrations are still high. We have really high amounts of RNA across the board. Liver giving us the highest amount with 25 micrograms of RNA total out of a 5 milligram sample and 44 micrograms roughly from the 10 milligram samples. And again, these RIN values fit well within the QC pipelines across the board. So here are the results from the 2100 bioanalyzer. Again, we have those really good RIN values displayed here, even with skin making it to the minimum QC threshold. One thing I'd like to add here is that all of these homogenization steps were taking place at room temperature. There's a lot of labs that really focus on keeping everything in cryo temperatures, and we wanted to display the results at room temperature just because of how effective a rapid homogenization feeding into a cold controlled RNA extraction. We kept all of our extraction process cold to try to maximize the RIN values on the back end here, and it really goes to show how well that works out. As far as keeping costs and extra added steps for your sample preparation low. And there's a lot of labs that want to homogenize these at cryo temperatures because they're worried about losing the integrity. But what we display here is that may not always be the case, especially if you're working with fresh samples. It may matter in other workflows, but specifically in this one, it was meeting the minimum QC threshold. Across the board, the fragments looked good, and all the RNA came out as expected. So in conclusion, the OmniBeat 96 in conjunction with the Omega MagBind DNA RNA kit provides high quality, high quantity DNA and RNA that meet QC thresholds for a variety of nucleic acid downstream analysis pipelines. DNA and RNA can simultaneously be extracted from a common sample. This provides a powerful starting point for multi-omic workflows and tissue models. And finally, high-throughput homogenizers paired with magnetic bead-based extraction kits create workflows that are easily incorporated into automation. This really helps labs that are looking to scale from maybe one sample into hundreds of samples at one time and are looking for a single way to extract both DNA and RNA from a common source to sort of keep the heterogeneity of the tissue at a minimum and give confidence in these workflows. With that, I'll turn it over to Nelson to speak more about automating this on liquid handlers.
Thank you, Caleb. I'm Nelson, and I'd like to introduce Omega's new MacBind DNA and RNA kit, which is compatible with automated workflows. This kit was designed to isolate both DNA and RNA from the same sample without having to split the sample. The main advantages of this kit are the increased sample efficiency. By using the same sample for both DNA and RNA, it is possible to use a smaller amount of sample. By extracting both DNA and RNA from the same cells, it helps to provide a comprehensive view of the genome and the transcriptome. It is compatible with the most common nucleic acid preservatives, such as RNA later, DNA and RNA shield, and others, with the relevant protocols already included in the kit's manual. It yields NGS quality nucleic acid with high DIN and RIN values and absorbance ratios. It is fully compatible with automation, which can provide consistency. This is an overview of the general workflow of the MacBind DNA and RNA kit. The sample is first homogenized with disruptor beads in RNA lock reagent, which helps to protect the RNA from degradation. A lysis bind master mix is added to the sample, which is then centrifuged. This is a one-step buffer, so there is no need to add lysis buffer and later a binding buffer in separate steps. The clear lysate is transferred to a new tube to which the magnetic beads are added, and then incubated at room temperature for the DNA binding. After the beads, which are now bound to the DNA, have been magnetized, the supernatant containing the RNA is transferred to a new tube and will be later used for the RNA isolation. The DNA isolation continues with the beads containing the DNA being washed twice, air-dried, followed by an elution and transfer of the DNA to the final elution tube or plate. The RNA isolation begins by adding an RNA bind mix containing more magnetic beads to the supernatant containing the RNA, which is also incubated at room temperature. After the beads, which now have bound to the RNA, have been magnetized, the supernatant is discarded. The beads containing the RNA are washed once and go through the DNA's digestion to remove any leftover DNA. The RNA is then bound to the beads again, followed by one last wash, air-dried, and eluted into a new tube or plate. This results in two separate elution fractions containing the DNA and the RNA. It's worth noting that the beads used for both the DNA and the RNA isolations are the same type of beads rather than different types. When automated the MacBind DNA and RNA kit, the initial steps will still require manual handling. The manual steps include homogenization, lysis buffer addition to the homogenates, and centrifugation. While the samples are homogenized, the instrument will add the magnetic beads to bind the DNA to a new plate, to which the user will then transfer the clear lysate after the centrifugation. This plate is then loaded back onto the instrument, which will now proceed with the bind, wash, and elution steps for the DNA isolation, followed by the RNA isolation without the need for further input from the user. The MacBind DNA and RNA kit is compatible with several different liquid handling platforms, such as Hamilton, Tecan, Dynamic Devices, etc. Although the MacBind DNA and RNA kit can also be used with magnetic processors, such as Kingfishers and the MacBinder, it will require more manual handling. This is an example of the deck layout for the Hamilton Star, which is capable of isolating DNA and RNA from 96 samples. The total runtime for processing 96 samples with a similar setup is around 3 hours. It's a very versatile layout, which can be customized to be used alongside other protocols, such as DNA-only or RNA-only isolation from other samples, or for other purposes such as library prep, cleanup, etc. The main components are a heater shaker and a position for the magnetic rack and plate, as well as tips and reagent containers. Here's some data we have obtained using this automated protocol. A link with a tech note containing all the data for the following tests will be shown at the end of the presentation and can also be found on the MacBind DNA and RNA product page at Omega's website. We use 10 milligrams of mouse brain and 10 milligrams of liver tissue samples, as well as 1 million cells for both manual and automated extraction. The tissue samples were homogenized using the Omni bead ruptor, and the clear lysates were loaded onto the Hamilton Star for the automated extraction. Downstream analysis for yield, quality, and fragment size were performed using a nanodrop, tape station, RT-qPCR, and qPCR. The yield and purity of the extracted nucleic acids were quantified using a nanodrop. The average DNA yields for both liver and cells was similar between the manual and automated extractions, and lower for brain samples when using automation. The average RNA yields was similar for both extraction methods for all sample types. The A260-280 and the A260-230 absorbance ratios were over 1.8 and 2 respectively for both extraction methods. These results are consistent with the yields observed during Omnis testing and also show consistency between the manual and automated methods. The fragment length and integrity of the extracted nucleic acids were quantified using a tape station. The DIN scores range from 7 to 9.9, and the RIN scores range from 6.2 to 9.2, depending on the sample type. High molecular weight fragments over 48 kilobases were obtained from the cultured cell samples. As mentioned by Caleb, the lower fragment size for brain and liver samples is likely due to the bead beating, which can shear the nucleic acids, lowering integrity values. So that's qPCR and RT-qPCR reactions were performed using 10-fold and 100-fold dilutions of extracted DNA and RNA to assess for possible contamination and inhibition. The average CTs were compatible between both manual and automated extraction methods, and the average delta CT between the dilutions was around 3, indicating no inhibition in either extraction method. To conclude, the MagBind DNA and RNA kit is capable of extracting DNA and RNA in separate fractions from a single sample, providing consistent and high-quality results. Both DNA and RNA are suitable for downstream applications such as PCR and NGS. This kit can be automated using different magnetic processors and liquid handlers, so please contact our automation team if you have any questions about automating this kit within your workflow. Thank you very much, and I'm going to hand it back to Claire.